Welcome to MSB Talks, a podcast about science for everyone. Our guest today is Dr. Greg Thurber, who is currently a professor of chemical and biomedical engineering at the University of Michigan. His lab uses in vivo microscopy and mathematical modeling to investigate the distribution of therapeutics from the organ level down to the tissue, cellular, and subcellular scale, with a particular focus on antibody drug conjugates for cancer treatment. And in this episode, we talk about Dr. Thurber's journey through his scientific background, research interests, and thoughts in the future of quantitative drug delivery and antibody-based cancer therapies. This is MSB Talks. We're glad to have you. And now for a conversation with Dr. Thurber.
Yeah, well, thank you. Thank you for agreeing to talk to us. Really means a lot and you know, helpful for our project. Yes, Arnav, do you want to give like maybe a quick background about what we do and then we can hear from you, Dr. Thurber, about what your research is as well?
Sure, that sounds good.
Yeah, sure. Well, yeah, first of all, thank you for agreeing to be on the podcast. It's great to be able to talk to a professor. Um, a lot of your work revolves around like antibody drug conjugates and finding molecular targets within the body for certain diseases, etc. We're using a similar approach. We're kind of focusing in on this idea of bacterial drug delivery. So we're taking a non-pathogenic bacterium. Um, One that's one aerotactic, so it navigates naturally to low oxygen environments, such as like a tumor microenvironment. Two, it's also like magnetotactic in the sense that if you like hold it up to an external magnetic field, it can navigate along field lines that you set. Um, and then to kind of guide it more specifically towards a tumor of interest, what we're doing is we're taking a ligand for a certain receptor that might be overexpressed in a given cancer, and we're essentially constitutively expressing that ligand within the bacterium itself. So, kind of three strategies to get a bacteria closer to where it needs to be. And then once it's there, we effectively just induce it to produce some kind of cytotoxin or payload. Um, but yeah, one way we've kind of thought of describing it is like an ADC, but without the linker. Effectively, you're bringing the bacteria to the site, and you're only producing the payload once localization has been confirmed.
Great. No, I definitely see a lot of similarities in terms of conceptually how the goals of your project are similar or parallel antibody drug conjugates for sure.
Yeah. Well, yeah, I guess we can dive right in then. Just straight into the podcast. So we've obviously read a lot about your lab, but if possible, could you tell us a little bit about your background in research, kind of how you got to where you are today?
Sure. So, when I was a student, when I was an undergraduate student, and even before when I was planning on going to um, college, I was interested in both math and science, and in particular medicine and engineering. And so I was trying to decide which career path to choose. And I was still debating between going to medical school or what I would do after that. So I decided to stick with engineering, and I took chemical engineering as uh, my major, and that allowed me to pursue both engineering. But on the side, I was taking a lot of biochemistry and molecular biology classes because I really enjoyed those classes. And then I think it was in my sophomore year that a professor, Wayne Curtis, approached me, or when I was talking with him about um, the classes, he was interested in whether I thought about research. And so I ended up working in his lab, and we were looking at producing therapeutic proteins. And so I really enjoyed that. I got to do some molecular biology, but there's also a lot of engineering involved. And so that's really what sold me on pursuing the path that I did. So then I decided to go to graduate school, and I went to MIT and talked with a professor there, Dane Wittrup, who developed yeast surface display, so a protein engineering technique for antibodies. And so this really gave me the opportunity to work on not just in the biospace and engineering, but really in medicine. And so I really enjoyed that. I had actually envisioned myself working in, say, a collaborative team in biotech, in industry somewhere. But then it was closer to the end of that time where he asked me if I'd considered academia. And I really enjoyed teaching. I really enjoyed my time as a teaching assistant. And so that's when I started to think about going into academia as a career. And I knew from the research standpoint that at the time we were doing mostly in vitro work with cells, but to push these molecules forward, we really need to demonstrate how they're behaving in vivo. So then I went to Ralph Weisslitter's lab at Mass General Hospital and Harvard Medical School to learn some of these in vivo techniques to track drugs. And that's really where I got into the molecular imaging side. And so imaging can be used both to in diagnostics as its own separate entity to screen for cancer or other diseases, but it can also be used to track therapeutics to help understand how they're behaving in design. And so that's really where then when I started at University of Michigan, that's really the combination of all these different factors. So the engineering of how these drugs distribute, how they're working in the body, and using some of the molecular imaging techniques to track them. So that's kind of where I ended up in the current position today, both in chemical and biomedical engineering here at Michigan.
Nice. And then, like, what would you say right now with your research or kind of just the field as a whole? What would you say is like the most exciting thing that you're working on or someone else is working on that you think has a lot of potential kind of within drug discovery and like molecular imaging?
Sure. So I would say there's probably three areas that we're very excited about. So, of course, antibody drug conjugates, this field has really exploded in the past few years. And I think, the field struggled for a while. We didn't fully understand as a field how these molecules were working. But now that we have a better sense of how to design them, and there's a lot of counterintuitive principles to how they're designed. So some of the things that might sound obvious and sound like they'd be beneficial end up not working very well. And we can get into some of those reasons later. And there's still a lot of um, misunderstanding in the field in many instances. And so that's one of the roles that I do to travel around and kind of teach people, especially those new to the field, how these molecules are actually behaving. But I think there's a lot of promise for ADCs. And in particular, we're very excited about how these molecules interact with the immune system and how we can better develop combination therapies to drive really strong immune responses that can help basically drive patients into remission for many years, even at later stages. So with the antibody drug conjugates, we're very excited about that field. Another field we're excited on the therapeutic side, and this is in collaboration with Peter Tessier here at Michigan, is in brain delivery. So they've developed a lot of efficient brain shuttles. So it's very difficult to get drugs into the brain. So similar to solid tumors that are difficult to target, the brain is another area because your brain tries to protect itself with the blood-brain barrier. But by developing these shuttles, they're able to efficiently get drugs into the brain. And so we're collaborating with that lab, which opens up a whole new realm of possibilities in terms of therapies. So I'd say on the therapeutic side, those are two areas we're very interested in. And then on the imaging side, we're excited about some of our recent work with non-invasive molecular imaging. So we just published a paper last year on a molecule that can be taken as an oral pill. So it gets absorbed into the bloodstream and binds to breast cancer. This is actually the second molecule we did this for. But the idea is for completely non-invasive molecular imaging and screening of breast cancer. So you can take these two molecules, they get absorbed in the bloodstream, bind to the tumor, and then you can use near-infrared light and just shine it on the surface of the body to detect whether there's a tumor there or not. So this can work really well for patients that might have very dense tissue where mammography doesn't work as efficiently. So that's one area we're excited about in the molecular imaging space.
That's a really interesting technique you just brought up
Yeah.
using minimally invasive imaging. I'm curious though, does that target like a certain receptor on breast tissues, or is it more like broad in its specificity?
Right. So we've developed two different targeting molecules, and they both complement each other. So they are both molecular. So some of the earlier work, you can actually, in the biomedical engineering space, some of the earlier work with near-infrared imaging was looking at non-specific agents. So you can actually, near-infrared light, if you take a red laser pointer and put it up to your fingertip, you can see it glow. It actually penetrates several centimeters through the tissue. So on the imaging side, this diffuse imaging through this tissue has been demonstrated in the clinic. But some of the initial clinical trials were on non-specific probes like indocyanin green that really you're measuring the physiology of the tumor, like the poor blood flow or the increased vascular leakiness. But with these agents, they target two different receptors. One of them is AV beta 3, which is present on a large fraction. So it's an integrin, and it's present on a large fraction of tumors and also tumor blood vessels. So this probe is really meant to increase the sensitivity of the method because it covers a lot of tumors and it has a very high sensitivity 800 nanometer dye on it. But we also want to improve on the specificity because there are other tumors that are benign. They're known as fibroadenomas that would also take up this probe. And so the second agent is binding to carbonic anhydrase 9, which is present in more hypoxic regions of tumors. And so it's more prevalent in malignant tumors that you actually have to treat. So one of the major challenges in breast cancer is the overdiagnosis and overtreatment. We know this just from statistics. The number of patients being treated exceeds those that have died from the disease in the past. So we know we're treating more than we have to. The problem is we don't know which patients we need to treat. And so that's really an area where better diagnostics, molecular information, as opposed to just anatomical tumors, we think could benefit.
Yeah, no, I think your approach there kind of rings a few bells with like what we're trying to do. I think it's a common strategy in precision oncology and precision medicine just to like look at any receptor that's overexpressed in a particular tissue type. For instance, like our system was initially set around ovarian cancer, which overexpresses folate receptor alpha. Um, so one of like the pillars of our project is using a peptide that's meant to kind of bind to this receptor, and then direct our treatment towards the tumor from there. Obviously on paper this sounds like, a foolproof plan, but in reality it's a little bit tougher to kind of validate this. From your experience, do you have any kind of thoughts on how best to optimize this?
Sure. So I think, one of the major aspects of any type of targeted therapy is really following the drug. And that sounds simple, but you can often end up with these surprises and counterintuitive approaches or counterintuitive results. So using ADCs or antibody drug conjugates as an example, early on in the field, there was a big push for kind of highly potent payloads. They're really toxic. They look good in vitro. But then once you go in vivo and you inject these drugs and look where they go, a majority of it doesn't end up in the tumor. So it gets concentrated in the tumor because of the binding. But if you just take a look at the total amount of drug administered, it mostly goes elsewhere. And so that leads to a lot of toxicity. And so then you end up with low doses. And the problem with the low doses, from the engineering perspective, is that they diffuse into the tumor very slowly. And the drug that does reach the tumor is actually poorly distributed because the binding rates are much faster than diffusion. So you end up with all the drug stuck right around blood vessels. Now, for your modality, you won't necessarily have to worry about that because you have an active, or at least depending on the strain of bacteria you use, you can have active chemotaxis. One thing that people don't necessarily think about is when we talk about antibodies, they're really passive targeting molecules. They get the same amount that goes into all the tissue. The reason why you concentrate it there is because of the binding. So you're slowing down the washout rate. So you can think of uptake into these tissues and just follow the rates and the concentrations of what you can achieve. So this is what I'd recommend for the bacteria as well. If there are studies published with similar molecules, how much is getting in the tumor, where is it going, and then what doesn't end up in the tumor, where does that end up? Because that can tell you what types of toxicity you might have. And those are really the two most important metrics. How do we get enough into the tumor and reaching the cancer cells versus the toxic dose to make sure you have that positive therapeutic window?
Yeah, and I guess like that, that's kind of like the like bystander killing, like you've got this toxic drug that can affect areas that are not kind of intended to be affected. But like, is that like all always looked at as just like a liability, or is it sometimes like could that be used somehow, like deliberately, where you target it at one area in the tumor, but it can kind of the bystander effect could just kill other regions around that are also tumor cells.
Yeah, absolutely. That's a great question. And again, I'll use an analogy from antibody drug conjugates, but I think there are parallels with many different types of targeted therapeutics. So in with ADCs, antibody drug conjugates, it's known as a bystander effect, where the small molecule payload, once the ADC is internalized into a cancer cell and releases that payload, depending on the physicochemical properties of that payload, specifically the membrane permeability, you may or may not get what's known as bystander effects. So if you have a small molecule that's fairly lipophilic and it can diffuse across plasma membranes, it can diffuse into nearby cells. Now, this is viewed as both, depending on your perspective, this can be a problem, right? So for many years, it was thought that we don't want bystander effects because we're targeting this to cancer cells. If it's releases the payload elsewhere, we don't want it to be able to get into healthy tissue. Now, there's a flaw in that argument, which I'll come back to. But let's first talk about the tumor. This can actually be very beneficial. And so, depending on the toxin you're talking about secreting from your bacteria, no matter what target you go after, whether it's folate receptor or really almost any other upregulated cancer target, there are often antigen negative cells, cancer cells that might have downregulated or mutated the receptor. So, how do you kill those? If you only kill the cells that express the target, you're going to maybe kill 99% of the cells, and then the 1% that doesn't express the target will regrow and your tumor will regress. But with a bystander payload, it can diffuse from an antigen positive cell into a nearby antigen negative cell and end up killing that antigen negative cell. So when you're going after these more heterogeneous tumors, these bystander effects can be very beneficial. Now, I just want to touch on that toxicity piece because everything with ADCs and a lot of these targeted designs is a double-edged sword. So there are molecules, and there are at least two approved agents in solid tumors that have payloads that don't have, or excuse me, not solid tumors, but two approved payloads that don't have bystander effects. There's more, but the two I'm mentioning are an MMAF version and a TDM1. The specifics of the payloads don't matter, but I want to point out that even though those payloads don't have bystander effects, they still have toxicity. So if the payload is released in the blood, yes, it won't get into healthy cells, but oftentimes these molecules are taken up non-specifically by healthy tissue. And when that happens, the payload that doesn't have any membrane permeability gets trapped in the healthy cell, and so it can accumulate. So actually having some bystander effect can help it diffuse out of that healthy tissue and can reduce some types of toxicity. So you really have to, this is where the molecular accounting comes in. You really have to see exactly how many molecules are going in what location, whether they're trapped there or they can wash out. And that's what ultimately dictates your therapeutic window.
Yeah, that was actually I kind of had fallen into that trap a little bit. I was under the impression that, achieving a higher specificity would be like a direct pathway to like a higher efficacy. But it seems like there is like a little bit of room for, a lower specificity leading to like a higher efficacy just through the bystander effect. Um, but one other thing you mentioned there was like this difficulty of penetrating solid tumors, with any kind of, therapeutic agent. Um, the type of cancer that we're addressing doesn't really rely on solid tumors, but one of our long-term goals is to adapt this therapy into something that can be used for various types of solid cancers. Obviously, I know that's a challenge kind of plaguing all of precision medicine right now, but do you have like any thoughts on how we can, kind of better navigate towards solid tumors and penetrate them?
Sure. So, I mean, I think using the example that you're developing, there's the possibility of active transport of these bacteria. And I would say that for many years, there was some research into bacteria for therapeutics. And if you go way back, there's the use of bacterial components as essentially an immune adjuvant. So using the bacteria, injecting it into tumors to drive an immune response. And that's still a possibility with the bacteria, even though you're looking at other types of modalities. But these bacteria, um, or excuse me, recent research has detected just endogenous bacteria that can populate some of these tumors. And so the fact that there are naturally bacteria in some of these tumor types really lowers the bar because people, and it's still a risk. You're going to be injecting bacteria into a patient that's already potentially sick. What is the safety considerations there? Uh, so those have to be weighed on the cost side. There's also the beneficial side where you can have active transport. I mean, you talked about the use of magnetic fields for targeting. Uh, that is one advantage that can be used in a primary tumor. It might not be as feasible in disseminated cancers, right? Where you might have hundreds or thousands of different micrometastatic deposits. Well, then it's hard to put a magnetic field locally around all of those. But in terms of chemotactic bacteria, if they're actually using energy to drive themselves towards some of these hypoxic regions, then you have an active transport to reach some of these areas. So that's one potential advantage, and that can also overcome other barriers. Cancer, or excuse me, tumors are not made of just cancer cells. Some of the more difficult to treat tumors like PDAC or pancreatic ductal adenocarcinoma. Pancreatic cancer is notorious for having very dense stromal tissue. And so it can be very difficult to get drugs at a high level and evenly distributed in those tumor types. So again, there's potential to help with distributing the drug that does get into the tumor by using some of these unique modalities. So we just published a paper led by Dr. Gulan Liu at Stanford and Eben Rosenthal's lab at Vanderbilt, looking at the distribution of antibody fluorophore conjugates in human tumors. And so you can see all the different barriers there are within the tumor microenvironment in these clinical tumors. So there's a lot of potential for these newer clinical techniques to better understand, what the barriers are so we can design more effective therapies.
Yeah, I mean, one kind of thing that you've been hinting at, would you agree that like imaging comes before like trying to like induce like cytotoxicity, like in terms of like creating a workflow for like validating an ADC? Um, like you mentioned, like antibody fluorophore conjugates. Do you think it's like necessary to kind of work through those and validate those before moving on to antibody drug conjugates?
I think it can be very beneficial. I think it really depends on the specific of the project. So there's been a lot of work in antibody drug conjugates, and so there are certain instances where we can design drugs fairly efficiently, even using computational methods, because of the amount of knowledge that's out there. And then you can go through some of the preclinical studies to just validate that this is behaving as predicted in the preclinical models, and you can predict what the clinical response rates will be. So for known targets, for known payloads, we've actually shown they're fairly predictable. Now that was again counter to what the field was thinking several years ago. But part of that is because of the challenges of animal models and how they may misrepresent the clinic. But we showed in one of our papers that if you use these preclinical models in with doses that are clinically tolerated, they're actually fairly predictive of what the clinical response rate would be. So if you have enough knowledge, you can do a lot of engineering ahead of time. For very novel approaches, there I would say imaging can be very useful to see exactly where your drug is going. I mean, even if you're secreting a uh, bystander payload, so the bacteria is secreting a molecule that can diffuse deeper in the tissue, that often is still not as effective as getting the therapy directly into the cell. And again, I'm using
Mhm.
ADCs as an example. Direct targeting of a cell is more effective than the bystander effect. So that's why getting your ADC evenly distributed can drive a much stronger response. So I'd say there would be parallels to other types of targeted therapies, like the project that you're working on, in making sure that you have a reasonable distribution of the agent that you have. And imaging is an excellent way to do that.
And I mean, you mentioned that like red light, like imaging. Would that be a like an imaging technique that you can use for that, or would other imaging techniques kind of be necessary to do the type of imaging that we would want?
Right. There's a lot of great imaging techniques. In our lab, we use a lot of near-infrared fluorescence microscopy. We like it for multiple reasons. There are lots of both fluorophores that you can tag different proteins, but there are also fluorescent proteins in the near infrared range that you could have, expressed by different bacteria. And near infrared light is useful because of the depth of penetration. Now, there's still a lot of scattering, so you don't have quite the precision that you do with other imaging techniques, but you can still use it. Let's say you have a subcutaneous tumor to detect the amount of uptake. But you can also do fluorescence microscopy. You can resect the tumor and look under the microscope to get the not only the whole tumor, but the tissue and the cellular level distribution. And one of the main
Mhm.
advantages of that near infrared light is the low autofluorescence. So there's very low background, which gives you very high sensitivity. So you can detect a very small amount of the drug. So that's why we use a lot of near-infrared fluorescence. We can do this multi-scale tracking. So biodistribution at the organ level and the blood level, and then hundreds of micron scale at the tissue, and then cellular and subcellular resolution. And we
Mhm.
also don't have to deal with radioactivity. So, PET and SPECT, these radiolabeled tracers, you can do excellent whole body imaging, but it's a little bit more challenging in terms of the safety. So, in an undergraduate program, I definitely recommend fluorescence. It's a little bit more feasible in terms of the safety aspects.
Yeah, like
Fair enough.
what we're trying to do is we're using a fluorophore that can fluoresce anaerobically. Um, it's called mini SOG. It just has like a flavin mononucleotide in its center as opposed to like the need for like oxygen catalysis. Um, so one like unfortunate part of our project is that we're constricted to only using cell lines. We're not allowed to use animal models. And as such, our plan is once we've kind of gotten our plasmid set up into our bacterium, that will co-culture it with an ovarian cancer cell line and then use confocal microscopy to kind of visualize if they are in fact co-localizing. Does that seem like a viable strategy? And if so, like what improvements might we make?
Sure. So I think there's a lot of approaches that you can take for this. Whenever you're doing a scientific project, you want to kind of build up from the most straightforward experiment to the more complex ones. So, some of the initial experiments that I would recommend just off the top of my head would be things like if you're expressing a targeting peptide on the bacterial surface, does that actually give you specificity? Is it binding to the
Mhm.
cancer cells? So if you have a tag on the bacteria, like a fluorescent protein, and you have this environmentally sensitive fluorescent protein, but you might also want to tag it with something that's constitutively on, that's on all the time, so you can detect it under any conditions. And that type of ratio imaging gives a lot of possibilities, right? So if you have your signal in the tumor, you can normalize it to the total number of bacteria. Because sometimes if you have a faint signal, you don't know if you have a lot of bacteria there, and it's just not, they're not in a hypoxic region, or you have very few bacteria, but they're in a really hypoxic region. So having multiple outputs can be beneficial. So, engineering the bacteria early on. But going back to some initial experiments, you can take the cancer cells and label them with the bacteria at different concentrations and see, do they actually bind? And if you have a cell line that doesn't express, let's say, folate receptor, do they bind less or not at all to that cell line? So those initial binding experiments can tell you, is this expressing properly? Are we getting the right binding? I wouldn't jump to this, but for the sake of time, just thinking ahead to some more complex experiments that you're doing, but not in vivo, there are a lot of really good organoid or tumor spheroid models that could be employed here, where if you grow those spheroids large enough, you can actually get hypoxia, and you could look at things like chemotaxis within this three-dimensional tumor microenvironment that still doesn't involve animal work.
Yeah, that's interesting because that's a similar technique that the last person we talked to, he's at U Chicago, but the last person we talked to had a similar idea of using organoids to mimic the that hypoxic region of the tumor. Because we're facing the same challenges. Um, I guess you talked about kind of the bacteria and like getting the receptors to bind. I guess one thing that the kind of modeling team has been working on is trying to figure out the right like balance between the bacteria being like retained near the tumor versus actually penetrating into it. And so, like, I guess how would you kind of think about that sort of trade-off? Um, and where do those sorts of targeting strategies tend to fail in your experience?
Sure. And let me just make sure I understand the question. You're saying how important is it to just get the total number of bacteria into the right location versus penetrating deeper into the microenvironment?
Yeah, I guess like they want to kind of find the right. I mean, they describe it as like stickiness essentially of the like the receptor binding or like the bacteria binding to the receptor, and like kind of trying to manage how much actually goes into the tumor versus kind of staying outside of it, but like around that area essentially.
Mhm. Sure. So, and this isn't something that you'd be able to replicate with the in vitro systems, but one of the things to keep in mind is when you're targeting a solid tumor, it's primarily through the tumor vasculature. So, once tumors grow beyond maybe a few hundred microns in size, they become hypoxic and they require recruitment of new blood vessels. And particularly for passive uptake of drugs like antibodies and ADCs, as soon as the tumors become vascularized, that starts to take over as the primary delivery route. If you think of diffusion from the surface of a large sphere, that diffusion is very slow. It is proportional to the length squared, and so it's really hard to get deep into the tumor. And
Hmm.
so primarily you're being released from the source. So now, many tumor blood vessels is an inflammatory environment. There's VEGF, vascular endothelial growth factor, that makes the blood vessels a little bit more permeable, so that can allow larger materials like bacteria to get into the tumor. But once they get in there, they might be stuck right around the blood vessels. And so I think, again, the details are important here. It depends on what your ultimate cytotoxic will be and whether that will be able to itself diffuse deeper in the tissue. But I think there would be some advantages of having, particularly if you're using bacteria that are chemotaxed into areas of hypoxia, there would be some advantages to leveraging that modality because ultimately, most cancer therapeutics are used in combination. And we have drugs, and it's a little bit easier to target perivascular regions of the tumor, more well vascularized regions. And so one of the advantages of having a motile bacteria would be the ability to get into some of those more hypoxic regions. So I would
Yeah,
say in terms of the advantages of your approach that you're taking, that would be a strength that you'd want to leverage.
that makes sense.
Yeah, cool. Um, but yeah, no, I think our computational modeling is pretty important, at least to how we're trying to visualize our project, because I think what they can do is effectively take our system and simulate it how it may work in systems that we can't replicate within the lab. So yeah, I definitely do agree that we need to kind of put as much effort into that as we can. Um, but
Sure.
yeah,
And
yeah.
one thing I will mention is our lab is a joint computational and experimental lab. A lot of the simulations we have, now we're not working with bacteria, but a lot of these molecules, like antibody drug conjugates, because of there's elevated interstitial pressure in the tumor because the blood vessels are leaky and they're not very permeable to fluid. But this means passive diffusion is a primary mechanism. And so you can actually have very robust predictions of where these drugs are going inside the tumor. And I think it's important to leverage that because there's a lot you can learn from the computational space. And then when you do go to spheroids or preclinical or clinical, it's more validating the predictions that you've made as opposed to starting from scratch and seeing what happens. So it's much more design than it is trial and error.
Yeah, I mean, I 100% agree. But yeah, no, I really appreciate all your technical advice and all the insight you've given. But yeah, I guess before we wrap up, we do like to ask all of our guests for a bit of advice as to, for anyone who's hoping to either be in their position or work in research or medicine or anything the like. But yeah, anything you could offer would be fantastic.
Sure. So maybe I'll give two pieces of advice. One would be on the technical side. As you're designing these experiments, make sure that the results are robust. That's one of the key features so that you can build on that. If you have some results that are a little unclear and you start to make assumptions on what's happening, by the time you get 2, 3 steps ahead, you might realize that those initial assumptions were incorrect and you might have gone down the wrong route. So making sure it's robust. And I also often tell students, particularly undergraduates in the lab, that when you run an experiment, it's going to fail. The most important thing is to have the right controls to figure out why it failed. So when you do it again, it can be successful. And so that perseverance aspect is extremely critical. And so that would be kind of on your thinking about next steps. So if you're interested in doing research long term, find something you love. It's challenging. You're doing something that no one else has done before. And so you want to enjoy what you're doing. You want to have that drive to overcome all the challenges that are inevitable in research. And if you do that, then you're going to do really well in the research field and you can continue to pursue your passions. So that would be my advice in terms of career-wise.
That's great. Thank you. Yeah, we really appreciate you coming on and talking to us. So yeah, Arnav, do you have anything else?
I think we're good. Just thank you again for giving us
Yeah,
the time to talk to you, and yeah, we'll hope to stay in touch about our project.
All right, absolutely. Well, yes, thanks for the discussion and good luck with the project.
All
thank
right.
you.
Thank you.
All right, take care.
You too,
Yeah,
Sue.
you too.
That concludes this episode of MSB Talks, where we delve into the science behind our research into cancer, therapies, and hope for a better solution. We're committed to sharing evidence-based insights and the voices of those advancing our understanding of cancer. And to stay up to date with future episodes, subscribe to our YouTube channel, and if you have feedback, questions, or personal stories you'd like to share, we welcome you to reach out. And you can find us on Instagram at @michigan.msbt. Speaking with Dr. Thurber were Arnav Sharma and Jack Collier, members of MSBT, and I'm your host, Daniel Yang, and I thank you for joining us today.